All right, so now we are in chapter 8.4, and we're going to talk about fermentation. So in the previous lecture, the 8.1 to 8.3 lecture, we spoke about aerobic respiration and anaerobic respiration. Recall that an organism is going to do aerobic respiration if it possibly can because it gets the most ATP, the most energy. If it's unable to do aerobic respiration, then it'll do anaerobic respiration. Remember, the difference is that in aerobic respiration, the oxygen is the final electron acceptor in the ETS, and in anaerobic respiration, something other than oxygen is the final electron acceptor in the ETS. However, in both of those cases, we require certain things. So in aerobic respiration, of course, we require oxygen. If there's not oxygen, we can't do aerobic respiration. We might do anaerobic respiration. But it also includes needing all of the genetic information for making all of the parts to do aerobic respiration, and also the genetic information to combat those oxygen radicals that are produced in aerobic respiration. In anaerobic respiration, then remember, we need to have some sort of inorganic molecule instead of oxygen. But in anaerobic respiration, we get less ATP formed in anaerobic respiration compared to aerobic respiration. Now, when we talk about fermentation, in fermentation, these are cells that cannot do respiration because of any certain thing, and we'll talk about some reasons. So again, aerobic respiration gets us the most ATP. Anaerobic respiration gets us less ATP, and actually fermentation does not get us ATP. What fermentation does, however, is it gets us the molecules that are necessary to make more ATP in glycolysis, and we'll talk about that in just a moment. So cells that cannot do respiration are going to be doing fermentation. And that means that some are permanently incapable of doing respiration. So like Streptococcus species cannot do either anaerobic or aerobic, so they're always fermenting. But then others, as we mentioned in the previous lecture, are facultative. And if they're facultative, then they can switch between different things, like perhaps aerobic, anaerobic, and fermentation, depending on the organism. So let's talk about reasons why we might do fermentation. So one reason is that the cell lacks the sufficient amount of appropriate inorganic final electron acceptor. Right, so we mentioned that it doesn't have oxygen available, so it won't do aerobic. If it doesn't have something other than that, so an inorganic final electron acceptor, then it can't do that either. So the cell lacks either enough or any at all of an appropriate inorganic final electron acceptor. So it won't be able to do respiration. Secondly, it lacks the genes to make the appropriate complexes and electron carriers, right? So if it can't even make the different protein complexes and the different carriers, then it's not going to be able to do the ETS. So it's not going to be able to do any form of respiration. So cell lacks the genes for appropriate complexes. I will say protein complexes and carriers. And then third, the cell lacks the genes to make any one of the enzymes in the Krebs cycle. So remember that that's part of respiration is the Krebs cycle. So we have glycolysis, the Krebs cycle, and then the ETS. And then if the cell doesn't have the ability to make the complexes for the ETS here, then it also, or it doesn't have the ability to make the enzymes for the Krebs cycle, again, it's not going to go through respiration. So cell lacks genes to make enzymes for Krebs cycle. So if it can't do those things, then it's going to go through fermentation. So again, some are incapable of respiration, so they only do fermentation to begin with. Others can switch. So remember that we said, we're going to write down the definition of fermentation here. So fermentation is the process that uses an organic molecule as a final electron acceptor to regenerate NAD+ from NADH. So that is the molecule that we're trying to get through fermentation. Again, fermentation is not producing ATP. It doesn't produce any ATP. what it does is it gets us back to kind of a starting point to then go through glycolysis to then make more ATP. So process that uses an organic molecule as a final electron acceptor. And this oftentimes is pyruvate, so I'm going to write that down here, pyruvate, to regenerate NAD+, which is what we need to go into glycolysis, from NADH. So glycolysis, can continue. So this organic molecule is typically pyruvate, and again, it does not, I'm going to write that down, so how about does not produce any ATP. So again, it's just getting us this NAD plus to then go back up into glycolysis and go through glycolysis again. So it does not have an ETS, so no electron transport system. It's just producing this NAD plus is the point of fermentation. So we have different types of fermentation, however. So let's take a look at, so the main one here is lactic acid fermentation. So we see lactic acid fermentation in lots of bacteria. So we actually have our lactic acid bacteria, which you know and we'll mention again. So in bacteria, but then also in our muscle cells, we have lactic acid production or lactic acid fermentation. So in our human muscle cells, when we run out of oxygen. So when we are using our muscles a lot and then we feel that kind of pain later, that's because of the buildup of that lactic acid, because of exerting our muscles and not getting enough oxygen to those muscles as we're exerting them. And then we start to go through lactic acid fermentation because we don't have oxygen to go through aerobic respiration. So we see that same thing happening with bacteria as well. So for example, our lab, which is our lactic acid bacteria. So some examples are lactobacillus, leuconostoc, and streptococcus. Oops. Streptococcus. So in this case, when we talk about our lactic acid bacteria, oftentimes, especially if you are familiar with lactobacillus, you may see this in things like probiotics. You may think of this as far as yogurt is concerned. And that's because this buildup of lactic acid actually causes the milk to solidify. It breaks down the proteins in the milk and causes them to solidify. And that's because what we're doing in lactic acid fermentation is we're taking this pyruvate. So recall that pyruvate is from glycolysis. We're taking the pyruvate and then we're taking the NADH that is produced. And remember, we're kind of recycling that NADH. And then what we're going to get is lactic acid and NAD+, so that this NAD+ can then go to glycolysis. And remember that the pyruvate came from glycolysis. So from glycolysis, we get the pyruvate, but we're not moving into the Krebs cycle. We're not moving into the ETS. Instead, we go through fermentation. So we have the pyruvate plus the NADH is going to turn into lactic acid and the NADH plus. So the NADH plus is then going to go to glycolysis so it can make more ATP. And then what we end up with is this buildup of lactic acid. So in the case of yogurt, for example, that lactic acid is going to then cause milk to solidify. And produce things like yogurt. And there are two different types of lactic acid fermentation. There is homolactic fermentation. And this is when lactic acid is the only product here. Lactic acid is only product of fermentation. So homo meaning same or only, and then lactic acid. So in homolactic, we're only getting lactic acid, which is what we've drawn up here already. Pyruvate plus NADH goes to lactic acid and NAD+. However, we do have heterolactic fermentation. Hetero meaning other, and in this case it produces lactic acid, of course, still, but it also produces ethanol and or acetic acid and carbon dioxide. So in this case, we get lots of other things. So hetero meaning others, so lactic acid, ethanol, and or acetic acid and carbon dioxide. So we see this when we see things like soured vegetables, so in producing things like pickles. So up here we're going to say an example. Example is yogurt. Down here, examples would be like pickles and sauerkraut. Things like that that produce that lactic acid, but they also produce other things. And that's why jars of pickles and sauerkraut, when they're fermenting, can burst, is because they do produce this carbon dioxide gas that can end up breaking the jars if you're not careful or doing it correctly. In heterolactic fermentation, we often also see they use the PPP pathway for glycolysis instead of the EMP pathway. So we have a lot of these other products being produced as well. We can say that also. So use the PPP instead of the EMP pathway for glycolysis. Okay. So our lactic acid bacteria, again, reduce the pH of the environment. They're making acid, lactic acid. And so this actually is helpful in many ways for restricting the growth of other pathogens. So when we are talking about fermentation, like for example, fermenting things like sauerkraut or pickles or even yogurt, we are decreasing the pH. It decreases the pH of the environment. And it prevents pathogens from growing. So in this case, when we're fermenting some sort of food product, it ends up being safe because we're only growing that particular bacteria, for example. And then it's going to restrict the growth of other pathogens. We see this also in the body, for example, in the vagina. So in there we have lots of lactic acid bacteria that are going through lactic acid fermentation, decreasing the pH of the environment there. And then what happens is it prevents other bacteria from taking hold and causing infections. Now, if that becomes imbalanced because you are not feeding those lactic acid bacteria the appropriate food, then there can be an imbalance and things like yeast can take hold and cause a yeast infection, or other microbes, other pathogens can take hold and cause other infections as well. So we see this helpful in many ways, this lactic acid fermentation. So the second one we are going to talk about is alcohol fermentation. And alcohol fermentation is pretty familiar, I think, to most people, along with the lactic acid fermentation, are the two main types of fermentation. In this case, we are producing ethanol. So produces ethanol. In this case, again, we're taking our pyruvate, but then instead, when we take our pyruvate, it's going to be converted into carbon dioxide and acetaldehyde. And then that acetaldehyde is going to be converted into ethanol. And this part right here, the acetaldehyde being converted into ethanol, is where we actually take this NADH and we're going to get the NAD plus from it. So again, remember that the goal of fermentation is to get the NAD plus. So in the end, our pyruvic acid or pyruvate is going to produce this ethanol here because we've taken the acetaldehyde and we produce ethanol, and we still get that CO2. So we get the bubbles from the production, the gas from the production, and we also get the ethanol from the production here, utilizing pyruvate. And then remember, the pyruvate came from glycolysis. And then this NAD plus is going to go back to glycolysis so that it can make more ATP. So then there is a table in the text that I think is helpful in comparing aerobic respiration with anaerobic respiration and fermentation. And so if you take a look at that table 8.2, you can see that in aerobic respiration, it gives an example, like in this case, it gives the example of Pseudomonas aeruginosa. Recall that the final electron acceptor is oxygen, right? Because oxygen is available, it's aerobic, meaning oxygen, aero. And then in this case, we're talking about EMP for glycolysis. We're talking about it then going into Krebs. Then we're going into ETS, including chemiosmosis, which we didn't talk about, but that's the actual movement, the gradient of hydrogen ions that are moving across the membrane that are then going back and producing the ATP. In that case, recall that when we are talking about aerobic respiration, we're talking about 36 to 38 ATP coming from that process. And then if we look at anaerobic respiration, remember that our final electron acceptor is an inorganic molecule that is not oxygen. So something like nitrate, which we talked about. We have other sulfur containing or iron containing end products here, carbon dioxide. So some examples that are given. And then the pathways that are involved in this, again, we're talking about glycolysis. So EMP, for example, again, going through the Krebs cycle, then going through the ETS, but of course the ETS ends with an inorganic molecule rather than oxygen. And then in this case, depending on the electron transport system that we're talking about here, we can get anywhere from 5 ATP on the low end to 36 ATP. So again, it's less on the highest end here than aerobic respiration, although it can be very, very low, all the way down to 5 depending on the situation. And then if we look at fermentation, our final electron acceptor is our organic molecule. In this case, typically what we're talking about is pyruvate. We're taking pyruvate, utilizing pyruvate. And then in this case, we can say if we're doing glycolysis, we'll just say the EMP pathway again. And then we're doing fermentation. So in this case, there's no Krebs, there's no ETS, we're just going to fermentation. And then recall that from EMP, we're getting 2 ATP total net, and from fermentation, we're getting 0. So our maximum ATP that we're getting from fermentation is 2, so very, very low. So these would be slow growers if all they can do is fermentation. But then again, this is why we see that a bacterium, if it can, will do aerobic. If it can't, anaerobic. And if it can't do either, then fermentation, because it gets the lowest amount. One last note about fermentation. In the text, there is a table that describes other types of fermentation. You should be familiar with these. So noting the acetone, butanol, ethanol fermentation. So in this case, we get acetone, butanol, ethanol, and then also the byproduct of carbon dioxide. And then this is important because we want to get things like acetone to use commercially, or butanol or ethanol, any of those things. We already know our alcohol fermentation, which is where we get ethanol. So ethanol, and then that's used for things like beer and for bread. In this case, of course, we're getting acetone, we're getting butanol, we're getting ethanol. Then we have our butanediol fermentation. In this case, we're getting formic acid and lactic acid. We also get ethanol from this, acetoin, butanediol, hence the name, some CO2, some hydrogen gas. In this case, this is what we use for wine, for example. So up here we can say beer and bread. This is our fermentation process for yeast going through bread. Here, wine is our butanediol fermentation pathway. We also have butyric acid. In this case, we're getting butyric acid. Wow. Butyric acid. We're also getting some carbon dioxide and some hydrogen gas. In this case, what we use this for is to make butter. Then we already talked about or spoke about our lactic acid fermentation, where we get lactic acid. And in this case, we spoke about sauerkraut, yogurt, cheese. Then we have our mixed acid, which we can get, from that we can get acetic acid, we can get formic acid, we can get lactic acid, succinic acids, ethanol, carbon dioxide, hydrogen gas, and in this case we get things like vinegar, and then some other pharmaceuticals and things like that. And then propionic acid is the last one. In this case we get acetic acid and propionic acid, as well as CO2. And this one is where we get our flavor of Swiss cheese from. So it's a different type of fermentation product. This propionic acid is what gives Swiss cheese its kind of Swiss cheese flavor. So you should be familiar with these types of fermentation as well. So that wraps up our fermentation section. You should know the difference between aerobic, anaerobic, and fermentation, why we use them, or why bacteria would use them, or why not.